Competent cells are specially treated bacterial cells that play a crucial role in genetic engineering, particularly in a process called transformation. In nature, bacteria aren't very good at taking up foreign DNA. However, in the lab, we can make them more efficient at this by creating what we call competent cells. These cells are prepared to incorporate external DNA, like plasmids, which carry genes we want to study or use. Competent cells are vital in molecular cloning, where scientists insert plasmids into bacteria to replicate and express specific genes. Without competent cells, this process would be much less efficient, making many genetic experiments impossible. One way to prepare competent cells is by using the chemical method. Here's how it works. First, we grow bacterial cultures until they reach the midlog phase, which is when the cells are actively dividing. We then treat these cells with ice-cold calcium chloride. This makes the cell membrane more permeable, allowing the DNA to enter more easily. Finally, we freeze the cells in small portions, making them ready for use whenever we need to perform a transformation. Another method is electroporation. In this technique, bacterial cells are washed in a special solution to remove salts. An electric pulse is then applied, creating temporary pores in the cell membrane, through which DNA can enter. After preparing competent cells, the transformation process begins. We thaw the competent cells and mix them with the DNA we want to introduce. Depending on the method, we either heat shock the cells or use electroporation. The cells are then allowed to recover in a nutrient-rich broth. Finally, they're plated on agar containing antibiotics. Only the cells that successfully took up the DNA will grow. Competent cells are essential tools in genetic engineering, enabling scientists to manipulate and study genes with precision. Whether through chemical treatment or electroporation, these cells make the complex process of transformation possible, paving the way for countless discoveries and innovations.